Welcome everybody to our webinar. My name is Oliver Rinner. I'm CEO of Biognosys, and today we present Dr. Loic Dayon, Proteomic team leader at the Nestlé Institute of Health Sciences. So the topic is pushing the sample size of biomarker discovery projects in clinical proteomics, and we will in a minute hear about that from Loic. Let me give you a very short introduction about Biognosys to start with. So Biognosys, our mission is to help people doing better biology by helping them to better understand the proteome. And I would say there's three main things we do at Biognosys. At the core of the company is our R&D. So we are inventors and developers of the most powerful proteomics methods that you can get. Working with key opinion leaders in the field to push the technology in terms of precision and depth, and as you will hear, also throughput, publishing this and develop new methods. And we make this available two ways. So people that are interested in proteomics but don't have access to mass spectrometers can access the Loic technology via our contract research services. So we get their samples and we design projects with our customers, be it protein expression profiling or assay development or also drug target deconvolution, and we help them to move their projects forward. For people that do have access to a mass spec, proteomics people themselves, we provide our software and reagent products so that they can implement the newest technologies in their own facility. So the reason why I find it especially interesting to have Loic here today is because I think he addresses a very important aspect in proteomics that will be important to push proteomics even further into use in the clinical research, for instance. So I think there's three main aspects where proteomics has made major progress in the past. So one is clearly the depth. The more proteins you see, the more pathways you cover, so depth is important. We are still all pushing that dimension. Now in tissues, for instance, one gets close to 8,000, 9,000, or even 10,000 proteins in a single sample injection with the newest technologies that are available. The other very important, I think, is the quality of the data. And here also proteomics has made huge progress from defining better statistical models, certainly supported by new instruments with very high resolution, so that the precision and the reproducibility of proteomics has really reached a level where it's feasible to use this as a very robust routine tool. But I think one aspect that has been a bit overlooked in the past is the throughput. So you can be very deep and very precise, but if this can only be done in 10 samples, like has been done in many scientific publications, then will never be useful really for large scale, for instance, clinical research. So it is necessary to move from low sample numbers, as they have been traditionally used for proteomics, to really high sample numbers. And here the technology or the field has also progressed a lot. And I think the work that Loic published and is keeping on developing is really pioneering this aspect. And I'm really looking forward to hear now from Loic about this just shortly. This is a webinar in a series of webinars that we do to promote the general proteomics, not only our technology, but proteomics in general, inviting several scientists. And if you look in our Science Hub, you find the recent recordings from our webinar, Rudi Ebersold, Andreas Bayer, Matthias Wilhelm, and others that we had here recently. But now I want to hand over to Loic, and I'm looking forward to his presentations.
Hello, everyone. So many thanks to Oliver for the nice introduction, and to Biognosys for the invitation to share some of our work on their Science Hub platform. So my talk today is entitled Pushing the Sample Size of Biomarker Discovery Project in Clinical Proteomic. And my aim with this presentation is to show you how mass spectrometry-based proteomics can be deployed in larger and larger research projects, and in particular in the clinical field. So there will be basically three parts to my presentation. So I will first describe the current status and rationale of the use of Proteomic in clinical research for the discovery of biomarkers. And as an introduction, I will go through the analytical challenges we are facing when we run clinical research studies for biomarker discovery using mass spectrometry-based Proteomic. Then I will show you a few tools that, among others, we consider as technological enablers to perform such larger studies. And I will finish with one recent application to identify novel diagnostic and nutritional concepts in the brain health field. So here on this slide, we can see the traditional route that has been used and is used today mainly for the protein biomarker discovery using mass spectrometry-based approach. This scheme can be referred as a funnel or triangular strategy. And initially, the identification of biomarker candidate is performed in a case control study with few samples. Here, typically tens. We then here we measure as many as possible protein analytes, and typically we aim at measuring 1,000 of protein in body-free tissue or cell extract. Then come the second part, and this is the verification base, which is based on the list of candidates. Targeted liquid chromatography tandem mass spectrometry is performed in an increased number of samples. The type of sample used at this stage needs to be compatible with future measurement in the clinic, and human blood is often preferred. And finally, the first stage is the validation of the remaining candidate, few of them, probably tens. And this has to be performed in thousands of samples using available technology. And in fact, when you look to this scheme, in fact, very few candidates make it through, somehow lagging the potential of Proteomic behind expectation. So several years ago, we decided to explore an alternative strategy. It consists in the Proteomic measurement of significant number of samples already at the very initial discovery phase. And this, we do it in a discovery untargeted manner. Our rationale here was that such an approach should provide increased statistical power, take better into account the population diversity, and finally reduce false positive and false negative hits. Such a strategy has been recently coined as rectangular approach by Dayon et al. In a recent review article. And importantly, it can be deployed from a cohort to another one. So here is an example showing you why sample size matters in Proteomic. And this is really obtained with real data. Here we use two large Proteomic datasets measured in our lab with mass spectrometry, which comprise about 400 and 600 plasma samples. And here we look very simply at the association of plasma protein between them in both cohorts. Then we did this considering randomly only part of the data, and we looked at the reproducibility of the findings. So let's have a look on the result on the graph. You can see that when you have low number of samples in both cohorts, found associations are fewer and not very reproducible. This is exemplified by the large error bars. Then when the sample size increases, these error bars decrease and the association could appear much more robust. So from this little exercise, we calculate that to obtain our association map, in fact, having less than 100 samples is not really sufficient. So now that we have exemplified in the previous slide that sample size consideration cannot be overcome, we need to see how realistic it is to apply it to mass spectrometry-based Proteomic for the discovery of biomarker in very large research projects. Ideally, as Oliver mentioned before, we would like to measure 10,000 protein in a sample, And this is indeed nowadays achievable when measuring cell extract with mass spectrometry. In a body fluid like plasma, it is much more challenging, in fact, to obtain complete proteome coverage, and this is mainly due to the high dynamic range of protein concentration in this sample type. Typically, today, we can identify several hundred of protein using a few hour analysis time with liquid chromatography mass spectrometry. For sure, we all know that the proteome coverage can be much more comprehensive in human plasma, but fractionation of the sample is required, and of course, as a consequence, the analysis time are much more longer. So in our lab, for example, we can, for instance, identify more than 2,000 protein in plasma, but typically we need 3 days to achieve such a coverage after sample fractionation. There is even other report of other groups that have broken some record, but to achieve such large coverage of the proteome in plasma, they basically needed a week of analysis time. So when you are thinking about large clinical project, of course, this long analysis time are not compatible, and we need to find some solutions. So to perform large proteomic study in clinical research, we still need to make compromise. And our solution was to limit the proteome coverage to allow increased sample throughput and therefore the analysis of a large number of clinical samples. And this is shown here on this radar plot. Here, no compromise is made on the quantitative performance of the measurement, nor on the other dimension of the radar plot. So along the long path to the generation of proteomic data and results, several factors are key for efficient analysis of large clinical cohort of sample. And one that we rapidly identify was the sample preparation, which is still often cumbersome in proteomics. Other are data management, data processing, on top of efficient liquid chromatography and mass spectrometry methods, those being label-based, label-free, or relying on data-dependent or data-independent methods. So both at the wet and the dry lab, optimized solutions are needed, and I will detail some of those in the next slides. So automated sample preparation appear to us very key when hundreds to thousands of samples need to be analyzed in order to increase the reproducibility of the results and reduce the risk of errors. So starting back in 2014, we developed a fully automated method for reduction, alkylation, and digestion of protein. And this robotic platform is also able to perform isobaric labeling, pooling, and sample purification. And here you can see the deck of our robotic platform. All the blocks, analytical blocks, can of course work independently of each other. So to validate the use of the platform, we compare its performance with what we are usually capable to obtain at the bench, and we found very, very similar results. But of course, the automated platform can deliver such performance day after day, weeks after weeks, and represents for us a very valuable tool of our laboratory. So in more details, our discovery proteomic workflow use filtration of sample, immunoaffinity depletion of 14 abundant protein, buffer exchange, digestion of protein, isobaric labeling, sample pooling, sample purification before reverse phase liquid tandem mass spectrometry. We first use it to analyze EDTA plasma, but we have also shown its compatibility with heparin plasma, serum, and cerebrospinal fluid. And here on the right, you have an example where we found very similar quantitative figure of merit for both plasma and CSF in terms of precision and accuracy. As well, we demonstrated in this evaluation the ability to obtain reliable calibration curve in a 2 proteome model experiment where basically we spike increased amount of an E. coli protein extract in a background of human plasma and CSF sample. So sample preparation is important, but it's far from being the only one enabler to the large clinical research study we want to perform in our laboratory. In those studies, sample tracking, management of information and data is very key. And here we have implemented a laboratory information management system, a LIMS, and developed a workflow capable of tracking the flux of information at each stage of the process, from the sample reception, aliquoting and storage, to the data acquisition and their processing. So for instance, during the sample prep, each step is recorded in our LIMS solution. As well, sequence of analysis for mass spectrometry acquisition are directly generated from the LIMS to avoid mistake and also to save us precious time. And finally, the database search, typically with mascots, and result gathering and validation that we do with scaffold is also launched and tracked automatically with our LIMS solution. So the last technical point I would like to talk today is about the processing of data. In our workflow, isobaric labeling is used for the quantification of protein, typically after the identification of those using a search engine like Mascot. And like many research groups, actually, we realized that lots of tandem mass spectra remain unexploited using this way of process. And interestingly, when we do this with isobaric labeling, this happened despite the presence of reporter ion from the isobaric tags in the spectra. So here we think that our dataset are generally far from being fully and efficiently exploited. And of course, this is even more true when analyzing a large number of samples, where the stochastic nature of data-dependent acquisition does contribute to the data missingness problem. So here we develop in collaboration an innovative solution that use machine learning tools, and we call it Quantify then Identify pipeline. It works with isobaric labeling type of data, and basically it reverses the classical order of steps for mass spectrometry data processing. The result is that, as you can see on the pie chart, we can exploit much more quantitative spectral data. And you can see it with the gray part, which is significantly reduced and represent the unexploited spectra. So here with this tool, we can now perform differential display on much more features, even if they are not yet matched to a proper peptide sequence. In addition to this tool, several modules are included. In particular, one allows to rescue missing data by further exploring the datasets and applying alignment and similarity score for the yet unmatched tandem mass spectra. Just note that we have not yet used this last and recent piece of work in a fresh clinical project, but we aim to do so. So altogether, those different technology development help us to perform proteomic at large sample size in clinical research. Other important elements not presented here and developed by other groups include higher throughput liquid chromatography and alternative mass spectrometry data acquisition methods. So just before going to the proper application part about the brain health project, let's have a look at some large data we can now obtain with these workflows. Here, we analyze the plasma of two large cohorts of 600 and 400 obese individuals. The second cohort was analyzed at 2 time points, making the entire study reaching almost 1,500 plasma samples analyzed. So here, very simply, we can look at the gender-specific protein, which appear here in color in the graph, and are differential between male and female. As you can see from the cohort to another, they are highly reproducible, and this shows the robustness of our measurement, but also that the rectangular design can be practically achieved using mass spectrometry-based proteomics in different and independent cohorts. So we think that getting this knowledge about the plasma proteome and its consistency, we can now use it and rapidly assess the quality of our proteomic datasets. So now I come to the application part. So in this second part of the presentation, I would like to talk about a project we do perform in collaboration with the local hospital of Lausanne in the field of brain health. Here we applied some of the proteomic technology I just described before. So as an introduction, you may know that Alzheimer's disease is the most common form of dementia seen in aging, and preventive practices still remain an unmet need. So the underlying pathology of AD seems to initiate 10 years or more prior to diagnosis, and this is shown in the graph here that displayed the model by Jack et al. Of the dynamic biomarker of Alzheimer's disease in CSF and in the brain. So here you can see that amyloid markers become abnormal first, then the biomarker of neurodegeneration, and they are followed later by the clinical symptoms. So here, importantly, early detection in the preclinical stage of dementia is essential to identify the individual at risk and try to intervene with preventive strategy like nutrition-based intervention. But in terms of diagnostic of Alzheimer's disease, it's still considered today as confirmed only by post-mortem examination of the brain. Identification of probable AD in vivo rely first on clinical examination that, as just said, happens too late and with the onset of the clinical symptom. Imaging techniques are also very valuable to provide early information, but they are still expensive and not available everywhere. And finally, specific measurement of CSF peptide and proteins such as tau protein, hyperphosphorylated tau, and amyloid beta can complement the clinical examination. They actually improve the workup and differential diagnosis, but they require quite invasive sampling. So clinical evaluation too late, imaging too expensive, and CSF measurement too invasive. So in this project, we studied a cohort of 120 older adults using proteomics and mass spectrometry technology. Here we follow an endophenotype approach, and we define our groups of individuals based on the level of well-known CSF tau and amyloid beta. As I shown on the previous slide, the alter level appears several years before the clinical onset of the disease. So our the cohort was separated into two groups based on the CSF level of phosphorylated tau versus A-beta ratio, which is representative of Alzheimer's pathology. Second, the cohort was separated into two groups based on the CSF level of A-beta, which is representative of amyloid pathology. So based on this group separation, here we have represented the demographic of this non-demented community-based population. And for both categories, we can see already some differences at age, mini mental state examination, and carrier of the APOE4 allele. So in this project, our main objective was to identify proteomic profile that can separate those groups and detect Alzheimer and amyloid pathologies. So this is the scheme of the design of the study. So in this study, we perform mass spectrometry-based proteomic in both CSF and plasma sample from those 120 individuals. Here we use the highly automated proteomic workflow and the list system previously described and collected the mass spectrometry data for data analysis. This is not the largest study we performed so far, but thanks to the tool we developed or implemented in the laboratory, the wet lab part of this study can be completed within a month of time. So let's first have a look to the CSF proteomic result. So in CSF, few proteins were differential between positive and negative a priori defined Alzheimer pathology in blue and in red, respectively, on the graph. And this was also true if you restrain the analysis to the subcohort of individual presenting cognitive impairment. So from this first assessment in CSF, we can already identify specific CSF proteome alteration that are related to Alzheimer pathology and may provide novel mechanistic insight. So to go deeper into this, we then decided to investigate association of CSF protein with individual most validated biomarker of amyloid pathology, neuronal injury, and tau hyperphosphorylation. And here are the results. So actually, what we found is that most of the correlation of CSF protein were with CSF tau and CSF p-tau, so phosphorylated tau, but not with amyloid beta, as you can see here on the cohort diagram. This result may suggest that CSF proteome alteration are more representative of tau pathology than amyloid pathology. The majority of those proteins were brain-enriched protein, including synaptic protein and protein involved in reelin-producing cell on the myelin sheath. We can see very strong association with tau and phosphorylated tau, new ones, as well as well-reported ones like neurogranin and neuromodulin here. However, if we consider the use of our findings for diagnostic purpose limited, given the nature of CSF. So next, I will review the result obtained with the proteomic profile of the 100 plasma sample. So from a diagnostic perspective, blood is very relevant sample. Its sampling is easily available, minimally invasive, and repeatable. Here again, we use exactly the same endophenotype approach to define preclinical Alzheimer and cerebral amyloid pathology with the aim of identifying plasma-based protein marker candidates. For the statistical approach, we use lasso logistic regression in order to select variable able to best classify our different outcome of interest. So first, we use the lasso approach to select variable traditionally available to the clinician. And second, we consider, in addition to this clinical variable, the protein measurement in plasma for the 120 subjects. And then in the third phase, we compare the result and the performance of both what we call the reference and the best model, including biomarkers. And this we do in terms of ROC curve and prediction accuracy. So for the detection of Alzheimer pathology, plasma protein did actually not improve the classification in the whole sample with respect to a reference model here depicted in blue. Then when the analysis is separately performed in the cognitively impaired people, the diagnosis accuracy of Alzheimer pathology was about 90%, with 19 plasma protein included. So here we observe a significant accuracy improvement here with respect to the reference model. And in particular, we think that this accuracy improvement may be particularly helpful in patients with cognitive impairment to identify those at risk of having cerebral Alzheimer pathology. As I said, blood-based diagnostic methods are non-invasive and easily available. And so they may be used to select patients, for example, for additional investigation with the more invasive or expensive tool, such as the lumbar puncture or imaging techniques. For the second endpoint of amyloid pathology, there are actually 16 plasma proteins that improve the diagnostic accuracy to 92%, and this within the full cohort. Here on the box plot are represented the level of each candidate protein selected with the lasso statistical approach. So in this case, identifying subjects likely to have cerebral amyloid pathology may be particularly important for recruitment in prevention trial at preclinical or very early clinical disease stage. So in this view, the diagnostic accuracy improvement from 80 to 92% using the plasma protein is quite encouraging. So in conclusion, our proteomic results in plasma suggest that several panels of plasma protein are relevant to CSF-defined Alzheimer and amyloid pathology. And the identified plasma protein may serve to improve differential diagnostic in cognitively impaired subjects, but also facilitate the identification of subjects at risk of developing Alzheimer's disease. Of course, at this stage, a replication in an independent cohort will be needed. And for this, we would aim at applying exactly the same identical proteomic approach. So with this work, in this work, the plasma proteome profile have been obtained on 120 individuals using mass spectrometry to guarantee what we think to be solid findings at the very initial discovery phase. And I think this is not something so usual and using a mass spectrometry-based proteomic approach. So what's next? So we consider here that we have additional opportunities, and we would like to further explore the obtained CSF and plasma proteomic datasets. And in fact, in this study, we quantified about 800 protein in CSF and about 400 protein in plasma sample. And when you look at the overlap, there is actually 256 protein which were identified in both compartments in pair sample of the 120 older adults. So our next perspective here is to study the relationship between the proteome of CSF and plasma and to evaluate their differences and the influence of the blood-brain barrier integrity. So at this stage, we have already determined several proteins with associated level between CSF and plasma. They are shown here. And we also shown that the blood-brain barrier integrity appears to be relevant, a relevant clinical measure to the proteome measurement and to their CSF to plasma ratio. But hopefully, I will tell you more about this result in a future presentation. So to conclude on the talk, I hope I convince you that larger and larger discovery proteomic study can be routinely performed today in clinical research, and that such an approach is indeed valuable. So of course, we still need to improve the technology, and in particular, we are still trying to increase the throughput of our methodology, the number of protein we can measure, and the data analysis method in order to better leverage the produced data sets that we have. So this is the last slide, the acknowledgement slide. So I would like to thank the member of my team, Ornella, Antonio, Charlotte, and Etienne, and our current and previous colleague at the Nestlé Institute of Health Sciences, Jai, John, Martin for the Proteomic, India, Eugenia, and Jean for the Brain Project, Francesco, Mona for the Bioinformatic, and Sergio, Irina, Armand, and Jorg for the Obesity Project. So they all contributed and supported our work over the years. So the work I presented today was rendered possible through several collaborations. The Brain Project presented its collaboration on a study led by Professor Pop and his team at the Centre Hospitalier Universitaire Vaudois in Lausanne. I also thank our collaborator Jérôme from Precision for Medicine. I do thank INSI Bio, especially Kostas and Seferina, for the bioinformatic development. And finally, many thanks to our collaborators, from the Diogenes Consortium and the Ottawa Hospital for the large study we performed previously in the obesity field. And I thank you for your attention, and I'm happy to take questions.
So thank you very much, Loic. And yes, now is still time for collecting questions. I hope you saw the chat tool in the GoToWebinar bar there. So please type in the questions that you have. And perhaps I start with some that already came in. I think directly to the talk. In the initial slide, you showed this association or the actual dependency of on the sample numbers. So what was again the readout? These associations. What is the measure of reproducibility depending on sample numbers that you showed there?
Yeah. So just a second. Can I go back to the?
Yes, I think you can.
Yes. Yeah. Perfect. This one?
Yeah, I think that's one. Yeah.
So here, basically, you have the number of samples which are considered in each cohort. So here we consider 2 cohorts, and what we here is the number of samples you consider when you make this association. So what you see, so let's take this example. When you take only 10 people in both cohorts, and you want, of course, to use one cohort and to confirm in another cohort, in the second one, you see that you find very few numbers of association, and the error bars, when you do it many times, because here we have a lot of sample to consider, is quite important. When you begin to increase the number of samples that you consider in both cohorts, then you see that first, the determination of the association is much more accurate. So you get more or less always the same numbers of association. And importantly, most of the time you find the same. And of course, the bigger you go with the number of samples, the best you are in reproducing your data in the first and the second cohort. So here the first cohort was 600 samples and the second cohort was 450.
Okay. Good. So then perhaps a bit related, a bit vague question, but I think it's still an important question. So giving the, say, the background variability in human plasma samples, so what's the good estimate of the number of samples that you actually need to do reasonable clinical studies? Probably depends, right? What is your ballpark?
It can always depend on the assay you use, of course. So in our lab, we are mainly use isobaric tagging technique, and to link the sample together, we have always a reference pool. But of course, this number need to be determined with the proper assay. But here, for example, on this data, if we want really to reproduce the association maps, we need 100 samples. I know it's not always possible. Sometimes samples are very precious, very difficult to get, but with our workflow applied on this sample, so the sample that we collect, because it can also depend on the collection of the sample, this is what we needed, based really on the data that we acquired.
Good, thanks. So then we move, to some technical question. And here's one that I think is obvious given the recent development in the field. So what's the pros and cons of using TMT method for biomarker discovery compared to DRA methods, in your opinion?
Yeah. So mainly, first we choose this workflow many years ago. So when we developed this platform, it was already 4 years ago. And basically what is important for us was to consider the throughput, and what we appreciate with the isobaric labeling is that the multiplexing capabilities. So meaning that when we run one sample, and actually this can be 6, 10 sample really behind. So in terms of the throughput, the throughput was really the driver of this, and this is why we chose this type of quantitative technique.
Thanks. And then also a technical question we get often as well is so how much do you gain by doing this extra step of plasma depletion versus non-depletion? Because obviously, with I guess the throughput is going down if you deplete.
Yeah, exactly. So this is the weak point of the workflow at the moment, because each sample needs to go through depletion. So it's one by one. This is so the most consuming step in the workflow. But actually, we still do it because we still get, let's say, 20–30% increase in the proteome coverage and the protein we can see today.
Okay. Good. And then here's a question. Okay, where is one again? Short ATMT measurements. Again, do you need to do MS to the 3? It's very technical. Perhaps you can shortly answer.
Yeah. So here we are on the good slide here with the two proteome model experiments. So here it's MS2, and you see first that what we got is reliable, in fact. But we also, of course, explored the possibility of doing MS3. And one drawback that we saw is basically the less precision we have on the results. So of course we are more accurate. We know that we measure better the ratios, but in terms of precision, it's not as good as what we can obtain with MS2, and probably because of the sensitivity of the MS3 extra step, I would say.
Okay. Then a question, you mentioned that the LIMS system behind the whole setup is very important. Is a question what kind of software it is and if it's freely available?
No, it's not freely available first. So for the moment, we are using Clarity LIMS. And we had to develop the workflow. So basically, you create your own environment, and yeah, it's really user-friendly.
Okay. So I think we covered the questions, and I want to thank you again very much for giving us this webinar. It's clearly very important to further push the ability to measure large sample cohorts with proteomics. I think I also found this slide very interesting, where you show really the impact on the quality of the readout depending on the sample sizes. And yeah, I hope that you all can move the field further in this direction.
Yes.
So thank you very much, Loic, and
thank you
very much everybody who listened. And have a nice evening or day.
Bye-bye.
Bye.